In the last few videos, we've taken a close look at the structure and the properties of our amino acids. In this video, we're going to be looking at how our amino acids are linked together to create peptides and proteins. Now, as we've seen previously, our amino acids can be linked together using the carboxylic acid and the amine functional groups on each of our amino acids. So these two functional groups can be linked together in a condensation reaction, as we see here. Right? So our carboxylic acid reacts with our amine. This is a nucleophilic acyl substitution reaction, and it creates this new amide functional group. This amide forms what we call the peptide bond linking our two amino acids together. Now, once we've linked two amino acids, we of course still have one carboxylic acid and one amine at either ends of our two amino acids. And so we can actually continue to link on additional amino acids here. This reaction can, of course, be reversed in a hydrolysis reaction. So that's our arrow over here. But under normal circumstances, this is a relatively slow reaction. And so we really need an enzyme to catalyze this if we're going to break our peptide bond apart and therefore break apart our protein or peptide polymer. Now, once we form our peptide bond, this actually slightly changes the behavior of our amino acid. So one of the things that we see with our amide here in our peptide bond is that we can have multiple different resonance forms here. So when we have our amide, so of course we have our lone pairs on our oxygen, as well as a lone pair on our nitrogen. Our oxygen, of course, is highly electronegative, and so it always wants to pull extra electron density towards itself. So we have a resonance structure where our electrons can get shared up to the oxygen, as shown with my two curved arrows here, and that creates my resonance form here with my negatively charged oxygen with 3 lone pairs, and my nitrogen using its lone pair to form a double bond to my carbon. We also, of course, have a third potential resonance form where the nitrogen keeps its lone pair while the oxygen takes the lone pair of the double bond. But this would be a very minor form because our carbon here doesn't have the full octet. Now, one of the consequences of this resonance here is that it creates a partial double bond between our nitrogen and our carbon. So rather than just looking at that as a single bond, it's really actually a partial double bond between our carbon and nitrogen, and then another partial double bond between our carbon and oxygen. Again, since oxygen is more electronegative than nitrogen, this is a reasonable resonance contributor because it does put the more of our electrons on that more electronegative oxygen. So it's not as large a contributor as the base structure over here on the left, but it is still a significant contributor to our overall hybrid structure. Now, one of the consequences of this for our peptides, as we're linking our amino acids together, is that we have a double bond that then is less flexible and less available to rotate. So as we're linking amino acids together, so here we have several amino acids linked together in what we call a peptide chain. So you can see here's one amino acid with our alpha carbon. We have that linked to a second amino acid, a third amino acid, and so on down our chain. Each of these carbon-nitrogen bonds are going to be, again, essentially rigid and prevented from rotating due to that partial double bond. So we'll have these rigid planes, essentially, linked together with our flexible bonds being only the 2 bonds to our alpha carbon. Right? So it's the alpha carbon to the nitrogen bond marked over here on the left, and our alpha carbon bond to the carbon of our carbonyl carbon. So all of our CN peptide bonds are not available to rotate here. So our amino acids then are linked together with these semi-rigid peptide bonds in order to form a variety of different polymeric structures. We refer to these as peptides if they're relatively small, or we refer to these as proteins if they're larger amino acid polymers. Again, all of our linkages here between our amino acids are going to be formed with these peptide bonds, marked here with this reddish outline. But of course, the individual amino acids can be any of our 20 different varieties of amino acids, right? So here we have 5 different amino acids linked together. You can see our varying side groups marked here in red. So we have serine down here at the end. You've got glycine, you've got tyrosine, alanine, and of course our leucine down on the right side. So again, we can change up what our peptide looks like just by changing what amino acids we have linked together. If we want to show the sequence of our amino acids within a peptide or a protein, we can do that using our single letter or 3-letter codes for our given amino acids. So this particular peptide here would be named as a SGYAL peptide based on the 5 different amino acids that are linked together here. Now notice that the 2 ends of our peptide here are unique from one another. At one end, we have an amine, where we could react and add on a new carboxylic acid. And at the other end, we have our carboxylic acid or carboxylate group. So every peptide or protein that we have will always have these unique amine and carboxyl terminated ends. We shorten these and call these the N-terminus, where we have our amine nitrogen. And we have the C-terminus, where we have our carboxyl. Every single polymer that we have made from our amino acids will always have that N-terminus and our C-terminus. Those are the free ends that haven't reacted in our condensation reactions. Our naming and, of course, numbering as well, because we do often refer to our individual amino acids with a number to tell us where in sequence that amino acid is. That numbering will always start from our amino terminus, that N-terminus, and it will go to the carboxyl terminus. So again, we can build up small polymers with our amino acids or much larger polymers. Both of these are very important within our biochemical systems. Peptides, or small polymers, have a wide variety of different roles. They're used as hormones, they're used as neuropeptides, antibiotics, and they're also used for protection, things like toxins in a variety of different species. As we get to our proteins, so again, the only difference between our proteins and our peptides is their overall size. So our peptides might have between 5 to 100 amino acids, whereas our proteins will have greater than 100 amino acids. As you can see here in our chart, some of our proteins will even have thousands of amino acids linked together in these extremely large, complex polymeric structures. Some of these also have multiple different proteins combined together into one structural unit. So that's noted here in the 3rd column over here. Now again, in order to create the wide diversity within our proteins, the diversity both in terms of structure and function, we need to have various amino acids. And these various amino acids, of course, are also combined together in various sequences in order to create these unique proteins. So for a typical protein, if we want to look at the full amino acid sequence, we might have something like this. So this is the actual amino acid sequence for one of our proteins that we find in the body. You'll notice it's just a long sequence of single letters. Again, each of these is representing one specific amino acid. As you can see, we have hundreds of amino acids linked together with a very unique sequence in order to create this protein. Any changes here in the sequence will affect the structure of our protein and will often affect the function of our protein as well. Now, one of the things that's unique about our protein polymers is that they form a very specific three-dimensional conformation or structure based on the sequence of amino acids linked together to form that polymer. And that specific structure or shape that our protein forms is key for it to fulfill its specific biological function. The normal regular structure of a protein we call the native or the native folded state of that protein. And normally what we see is that a protein has to fold into that specific shape in order to be active. And if we disrupt that shape, we will disrupt the activity of the protein as well. So how exactly do we go from a simple linear chain of amino acids to a very complex 3D structure? Over here on the right, you can see a representation of a protein 3D structure. And notice how complex this surface is, right? We have all these bumps and indentations within the structure that then represent different places where a protein can potentially interact with other biological molecules. So it's very complex to take just a sequence of amino acids and figure out the one specific way that that's going to fold together to create a 3D shape. Even though our proteins have such a complex 3D shape, many proteins are able to find that one correct native folded structure just based on the individual interactions between our amino acids, arranging themselves into that correct structure, often without any added assistance. Now, in order to understand the full picture of our 3D protein structure, we normally break it down into four individual components. We have our primary structure, which is just the sequence of our amino acids, right? From, again, the N terminus, where we have that free amine group, to the C terminus, where we have our free carboxylic acid. We have our secondary structure, which is the local organization of our amino acids. Our tertiary structure, which is the overall 3D structure, once we've taken all of our different smaller structures and combined them together. So you can notice individual smaller structures kind of combined in this larger 3D shape. And then we have quaternary structure, which is formed when we take multiple different 3D proteins and combine them together into a larger protein complex. So we're going to take a detailed look at each of these levels of protein structure in order to understand how proteins form into these specific structures. So we'll start off with secondary structure, since we've already looked a little bit at our primary structure and what the different amino acids are and how they behave. So when we're forming secondary structures, what we're looking for is we're looking to maximize favorable interactions and minimize unfavorable interactions. Really, this is what we're doing for all of the different levels of protein structure. We're looking to form as many favorable interactions between our amino acids while eliminating unfavorable interactions. As we're forming our secondary structure, the primary thing that we're going to be looking for is hydrogen bonding. So as we bond our amino acids together, we of course always form our peptide bond, which of course is an amide functional group, between each of our amino acids. Now within our amide, we have of course our hydrogen bond donor of our carbonyl, or this is our hydrogen bond acceptor rather, and our hydrogen bond donor of our nitrogen down here. And so these peptide bonds will actually be able to interact with other nearby peptide bonds through hydrogen bonding. So as we're looking to form that local structure, we're going to be looking to maximize the hydrogen bonding between our peptide bonds, right? So we want all of our amide functional groups to be hydrogen bonding within our local structure. Now, the other thing that we're looking to avoid as we are forming our local structure is unfavorable steric crowding interactions. Now, remember, as we saw earlier, right, with these two resonance structures of our peptide bond, our peptide bond is actually relatively rigid. So we're looking to kind of fold up this polymer, but it's a polymer that's made of rigid individual pieces. So we're really just looking at a few different angles here of these bonds can rotate in order to try to form those hydrogen bonds. So we have some limited range of mobility, and at the same time, we also want to avoid any steric clashes as we bring any of our groups too close together, right? So as always, as any two atoms come too close together, we get steric repulsion from the electrons around those atoms. And so we can't just bend our chain up into whatever shape we want, right? Some shapes are going to result in atoms pressing up against each other and repelling away from each other. So this rigid shape of our peptide backbone is going to somewhat limit the shapes that we can take with our peptide chain here. So we're really just looking at these two key angles around each of our individual amino acids. We have the phi angle, which is the angle between the alpha carbon and the amide nitrogen. And I like to remember this by using the mnemonic fin, right? So if you think of like a fin from a fish, right, that tells us that the phi angle is the one that's our alpha carbon connected to the nitrogen, right? That N corresponds to the N of our nitrogen. Our second angle here is the psi angle, which of course is the angle between the alpha carbon and the carbonyl carbon. These are really the only two key angles that we have to worry about as our polymer is rotating around and bending itself into different shapes. Again, that's because the peptide bond is essentially rigid and linked in just that single rigid plane. So for the psi angle, in order to remember that that's corresponding to the alpha carbon and carbonyl carbon bond, I like to think of this as the Sic angle, again with that C there corresponding to the C of my carbonyl carbon. So that's just a little shortcut that I use to try to help remember. Phi angle corresponds to the nitrogen bond. The psi angle corresponds to the carbon bond. So here we have these two phi and psi angles that essentially determine the overall shape of our peptide sequence. Again, some of those phi and psi combinations will be very unfavorable because of steric crowding between our backbone atoms or other atoms within the side chains of our amino acids. Some of our phi and psi combinations are going to be more favorable because of the chance to form those favorable hydrogen bond interactions between our peptide bonds, right? Between that carbonyl hydrogen bond acceptor and our nitrogen-hydrogen hydrogen bond donor there. So these are the two key principles that we'll see repeated over and over again in all of our different secondary structures, right? It's always just balancing the need to avoid steric crowding and the need to form favorable hydrogen bonding. In order to determine what phi and psi angles actually are favorable, the Indian scientist G.N. Ramachandran used computer models, and he looked at he manipulated each of those phi and psi angles and looked at when there would be significant steric crowding between our atoms here versus when there might be more favorable hydrogen bonding. So using that computer model, so he's just bending each of those angles and looking at when you start to see crowding, he was able to develop this Ramachandran plot. So here's what a Ramachandran plot looks like. And essentially what this plot says, it's just showing the two different angles here. So we've got our phi angle and our psi angle here on the y-axis. And we have all of our possible angles from -180 to +180. So that's a full 360 degrees of rotation around those particular angles. And these areas in blue here are areas where there isn't significant steric crowding between our atoms as we're rotating around those bonds. And as it turns out, these are the areas where our secondary structures will sit, right? So they'll always use these particular angles where we don't have that crowding between our atoms. There are a few rare exceptions, but in general, all of our common secondary structures will use these key angles. So again, our secondary structure is that local rearrangement of our polypeptide backbone. And the two regular arrangements that we commonly see are the alpha helix and the beta sheet. And again, both of these will use psi and phi angles that will minimize that steric hindrance that we saw plotted in that Ramachandran plot. We do also see some regions that will be have an irregular arrangement, so they won't have a specific secondary structure, and we call these random coil regions. So for our alpha helix and our beta conformation, so we can see here the actual phi and psi angles that we commonly see. So the alpha helix will have this -57 on our phi angle and -47 for our psi angle. You can see we have specific angles that we'll commonly see for our beta sheets. And we also have some other kinds of secondary structures down here. Each of those will have specific angles that determine what that shape normally looks like, right? So those angles will set our amino acids into a specific conformation, and that will be repeated over and over again in this regular secondary structure. So again, we can use those angles to actually point out our individual secondary structures on our Ramachandran plot. So you can see our alpha helices are either in this blue region down here, or we have this little region up here. This is for more rare left-handed alpha helices, which we'll talk a little bit more about later. We have all of our different beta sheet varieties all right up here in this blue region, as well as some other interesting secondary structures, such as the triple helix that we see in collagen. Again, we'll talk about each of these in more depth later. I just kind of want to show you generally how that Ramachandran plot, again, corresponds to what types of secondary structures we normally see, right? Again, we have specific phi and psi angles that we can take for our amino acids, and those will correspond to specific regular organized secondary structures within our overall protein or peptide structure.